The banded newt in Israel, Ommatotriton vittatus distribution, habitats, life cycle, reproduction, and genetic variation between different phases. Writing the film's transcript and scientific editing. Gad Degani. The banded newt, scientifically referred to as Ommatotriton vittatus, previously classified as Trichuris vittatus, is one of 3 species within the Ommatotriton genus, alongside Ommatotriton nestorovi and Ommatotriton ophidiacus. These species are distributed across Turkey, Syria, and Israel, showcasing impressive adaptability to highly variable environmental conditions. Notably, Ommatotriton ophidiacus and Ommatotriton vittatus differ in several aspects, including the number of trunk vertebrae, genome size, and allozyme patterns. Ommatotriton ophidiacus, the most northernly distributed species, is divided into 2 groups: the western group, inhabiting western Anatolia in Turkey, and the eastern group, found throughout the rest of Turkey and the western Caucasus. This genus comprises 3 species: Anatolian newt, Caucasian newt, and the southern newt. The southern newt may consist of more than 1 species. Isolde van Riemsdyk et al. 2021. Most common knowledge about the newt is about its short life cycle in the water part as an egg, a tadpole, and its life in water. Why? It is difficult to study the newt after it is metamorphosis and lives on land. The breeding sites span habitats that have been studied. In Israel, newts are found in relatively dry conditions. The climate in Israel varies from the lush areas in the north down to arid conditions in the south. We find bend newts around bodies of water, those that are unstable like a pond or a waterhole. The life cycle of the banded newt, Ommatotriton vittatus, in Israel can be described as follows. During the first rains of November-December 2020, adult male and female newts migrated to ponds that had not yet filled with water, seeking refuge under stones for approximately 1 month under challenging environmental conditions. December 27, terrestrial female and terrestrial male. About 2 weeks after the ponds filled with water, the newts transitioned to their aquatic phase, during which sexual behavior and mating occurred. February 15, aquatic male, aquatic female, and mating and fertilization.
Newt's eggs were first observed on these plants 1 month after the ponds filled with water. Between February 15th and March 14th, 2021, eggs and embryos were documented on the common water crowfoot. In early March, freshly hatched newt larvae were observed, and by March 21st, only small, newly hatched larvae were present. March 7th, egg. March 14th, embryo. March 21st, larvae. May 25th, terrestrial juveniles.
Sexual behavior and transfer of sperm, spermatophore during the breeding season. Male banded newts exhibit elaborate courtship behaviors in aquatic habitats. They perform visual displays such as waving their tails to attract females. Once a female is receptive, the male deposits a spermatophore, a packet containing sperm, on the substrate. The female picks up the spermatophore with her cloaca, ensuring the fertilization of her eggs internally. Egg laying on aquatic vegetation. Fertilized females lay eggs individually, carefully attaching them to underwater leaves or other vegetation in the water. This behavior provides the eggs with protection from predators and environmental disturbances. Each female can lay several eggs over a period of time.
The development of the banded newt larvae begins with the embryo undergoing a transformation into a larva. During this stage, the larva develops external gills on the sides of its head. The embryo is still surrounded by its amniotic membrane while it moves within the egg. Eventually, the larva hatches from the membrane and begins swimming freely.
This chart shows us the correlation between the genetic variations and the different locations. R square equals 0.706, p less than 0.01. Here we see, in the Upper Galilee, among the grazing areas for cattle and horses, the Nahalit pool. Unpredictably, it only appears in the winter and early spring. Differences in gene expression were found between newts on land, newts in aquatic settings, and larvae. 1. Terrestrial adults newts, males and females. 2. Aquatic adults newts, males and females. 3. Aquatic larvae. After analyzing tail tissues at different phases, we found clear and distinct 1. Terrestrial newts, males and females. 2. Aquatic newts, males and females. And 3. Tadpoles before metamorphosis. The groups were clearly defined by hierarchical tree analysis. 1. Terrestrial adults newts, males and females. 2. Aquatic adults newts, males and females. And 3. Aquatic larvae. The largest number of D genes was found between tadpoles and adult aquatic newts. 7K7.4 D genes. Comparing the aquatic and terrestrial adult groups showed 2.4 D genes. Numbers of D genes in each comparison. Venn diagram of genes that were D in one or more comparisons between the groups. This is a chart showing the number of gene D in each group. Using HCl heat map, we found similar results. Once again, the expression of the genes in larvae is different from the other groups. Larvae, aquatic F and M, and terrestrial M and F.
Differentness exertion. Down regulation. No sin. Up regulation. Differentness exertion. Down regulation. No sin. Up regulation. Differentness exertion. Down regulation. No sin. Up regulation.
A proposed qualitative model highlighting the link of selected genes and pathways to different phases in the life cycle of Ommatotriton vittatus.